Hi everyone, welcome to Microbiological Insights. So today in this video we are going to see about another important topic in the microbiology subject, that is the preparation of microscopic slides for the staining of specimens. What's the need for staining the microbial cells? Because the microorganisms are colorless in nature. So if we want to visualize the colorless microbials through the microscope, it is not possible. So in order to visualize the microbial cells clearly, we need to stain the microbial cells. The staining is not a single step process, so it is a stepwise process. Before staining, we must fix the bacterial cell in the microscopic slide. This preparation helps us to increase the visibility of the microorganism. And another important feature is this process also makes the specific morphological features of the cell to visualize it with more prominent. And another important one is that in order to preserve the slide or the microbial organisms for the future study, we need to fix and stain the cells. So in this video, we will see both the fixation process and the dyes which is used for the staining methods in a detailed manner. Now we will see the fixation process in detail. What is fixation? Fixation means fixing the microbial cells in the microscopic slide. Fixation is the process by which the internal and external structures of the cell and microorganisms are preserved and fixed in position. So what happened during this process? In this process, the internal and the external structures of the microbial cells are preserved and it is fixed in a particular position. So during staining, the cells are preserved and it cannot be washed through the action of the particular staining dyes. And another important thing is it inactivates the enzymes that might disrupt the cell morphology and toughens the cell structures so that they do not change during staining and observation. Another important role for the fixation process is that it inactivates the enzymes that might disrupt the cell morphology. So some enzymes disrupt the cell morphology. The fixation process inactivates that particular enzymes and toughens the cell structures. So the cellular structures are toughened and it cannot change during the staining and observation. So during staining, the cells are fixed in a particular position. And another one is that during the fixation process, the microorganisms are usually killed and attached firmly to the microscopic slide. So we cannot visualize the live cells during the staining process. So before staining, we need to kill the microbial cells and fix it in a particular slide. There are two types of fixation. What are they? They are the heat fixation and chemical fixation. Heat fixation means we are fixing the microbial cells through heat. So this is the routinely used process to observe the prokaryotic microorganisms. So for prokaryotic microorganisms, we are using this fixation method. In the heat fixation process, typically a film of cells or a smear of cells is gently heated and it is passed through a flame. So we have to prepare the microbial smear and then we need to pass the microscopic slide through the flame to fix the microorganisms. The heat fixation preserves the overall morphology but not the structures within the cells. The heat fixation preserves only the cell morphology, overall cell morphology, but it cannot preserve the internal structures of the cells. The next one is the chemical fixation. Chemical fixation means we are fixing the cells by using the particular chemical. The chemical fixation is used to protect the fine cellular substructure and the morphology of the larger, more delicate microorganisms. So to protect the fine cellular substructure and morphology of larger delicate microorganisms, we need to fix the microorganisms using a chemical. What is a chemical fixative? The agent or the chemical agent which is used to fix the microbial cells. So these chemical fixatives penetrate the cells and react with the cellular components. What are the cellular components? Proteins and lipids. So this chemical fixatives penetrates inside the cells and react with the proteins and lipids and make them inactive, insoluble and immobile. So it is fixed in a particular microscopic slide. So then the common fixative mixtures contain such components as ethanol, acetic acid, mercury chloride, formaldehyde and glutaraldehyde. The chemical fixative contains what are the components? Ethanol, acetic acid, mercury chloride, formaldehyde and glutaraldehyde. This is the schematic diagram of the heat fixation. So in the first microscopic slide, we are preparing the inoculum using the inoculation loop. So we need to smear the microorganisms in a thin film. Then after preparing the thin film of the microorganism, we need to air dry the microorganisms, and then after air dry only we have to pass the slide through the flame to fix the microorganisms. The next one is the dyes. The many types of dyes used to stain the microorganisms have two features in common. So there are many dyes which are used to stain the microorganisms, but the most of the dyes which we are used for staining have the two major features in common. The first one is they have the chromophore groups. What is a chromophore? Chromo means color. So color producing groups are present in that dyes. Chromophore is nothing but a chemical which is used to stain the microbial cells. And the chromophores also have a conjugated double bond group. The conjugated double bond group present in the chromophores only give the dye its color. So because these chromophore groups bind with the microbial cells with the help of the ionic, covalent or by hydrophobic bonds to fix the to apply the stain to the microorganisms. Another important character of the dye is that most of the dyes are used to directly stain the cell or the object of interest. But some dyes such as India ink and nigrosin are used in negative staining where the background but not the cell is stained. The unstained cells appear as bright objects against a dark background. So most of the dyes nowadays we are using directly stain the microbial cells. But some of the dyes such as the India ink and nigrosin stains only the background and not the cells. So when we are using the India ink and nigrosin dyes to stain the microorganisms are not stained. So during negative staining we see the microorganisms in the dark background. The cells are unstained. The dyes that bind cells by ionic interactions are probably the most commonly used dyes. I already told you that the chromophore groups bind with the ionic, covalent or hydrophobic bond interactions. So normally the dyes that binds by means of the ionic interactions are probably used as a most commonly dye. So these ionizable dyes may be divided into two general classes based on the nature of their charged group. What are they? The basic dyes and the acidic dyes. What are basic dyes? Basic dyes have positively charged groups, usually some form of pentavalent nitrogen, and are generally sold as chloride salts. So the basic dyes are generally sold as chloride salts, and these have the positively charged groups. What happens when we are applying these basic dyes to the cells? The basic dyes bind to the negatively charged molecules such as the nucleic acid, proteins and the surfaces of the prokaryotic cells, because the basic dyes are positively in nature. The bacterial cells such as nucleic acid, proteins and the other surfaces are have negative charge. So The positively charged basic dyes bind with the negatively charged nucleic acid proteins and the surfaces of the prokaryotic cells. What are the examples of the basic dyes? Methylene blue, basic fuchsin, crystal violet, safranin, malachite green are some of the examples of the basic dyes. Next we move on to the acidic dye. From the name itself you understand that acidic means it has acid groups or hydroxyl groups. Acidic dyes possesses negatively charged groups such as carboxyls and phenolic hydroxyl. So the acidic dyes have negatively charged groups. What are the groups? The carboxyl and phenolic hydroxyl groups. Then the acidic dyes possesses the negatively charged. So it binds to the positively charged cell structures. The basic dyes have positive charges, so it binds to the negatively charged cell structures. Here the acidic dyes have negative charges, so it binds to the positive charged cell structures of the microbial cell. What are the examples of acidic dye? Eosin, rose bengal and acid fuchsin are some of the examples of acid. Next, some of the properties affect the ionizable dye's effectiveness. What are the properties? The staining effectiveness of ionizable dyes may be altered by pH since the nature and degree of the charge on cell components change with pH. So, the effectiveness of the ionizable dyes such as the basic and acidic dyes may be altered by pH because the degree of the charge on the cell components of the microbial cell components change with pH. So, the effectiveness also altered by changes if any changes occurred in the pH. Thus, acidic dyes stain best under the acidic conditions when proteins and many other molecules carry a positive charge. So, the acidic dyes stain best under the acidic condition. Acidic dye means it can stain best under the acidic conditions. At that time, the proteins and many other molecules have positive charge. And the basic dyes are most effective at higher pH. The dyes that bind through covalent bonds or because of their solubility characters are useful. So, the dyes that bind to the covalent bonds. What are covalent bonds? The chemical bond that involves the sharing of electron pairs between atoms. That is termed as the covalent bond. So, if the dye bind through the covalent bond or because of their solubility characters of the dyes, they are useful for the stain. For example, the DNA can be stained by Fulgen procedure in which the staining compound covalently attached to its deoxyribose sugars. So, what happened during that staining process? The Schiff's reagent is covalently attached to the deoxyribose sugars of the DNA molecule. And the next example is this Sudan 3 or Sudan black. This Sudan 3 dye selectively stains the lipids because the lipid is soluble in nature but it does not dissolve in the aqueous portion. So, the Schiff's reagent is covalently attached to the deoxy sugars of DNA and the Sudan 3 selectively stains the lipid molecule. I hope everyone understands this topic. If you have any doubt regarding this subject means you can write your doubts in the comment section. If you like this video, please like, share and subscribe my channel.
If you like this video, please don't forget to subscribe, share, like and click the bell button. If you have any more doubts, you can write your doubts in the comment section below. Thank you.